Good afternoon to all audience. I, Dr. Pushpendra Kumar Jain, Dean IIMT College of Pharmacy, would like to share my knowledge about ICH guidelines among all of you. So let's start discussion. First question comes in mind. What is mean from ICH? ICH is a unique project that brings together the regulatory authorities of Europe, Japan, and the United States and experts from the pharmaceutical industry in these three regions. Why choose these three regions? Because the new drug discovery is very fast in these three regions. What is the need of experts from pharmaceutical industry in this committee? They are responsible to discuss all the scientific and technical aspects of product that to be registered in these three regions for marketing or production or any kind of purpose. Name of ICH is International Conference on Harmonization. Aim of this conference to give the technical requirement for the registration of pharmaceutical for human use. This harmonization was established in 1990 as a joint regulatory or industry project to improve through harmonization the efficiency of the process for developing and registering new medicinal products in Europe, Japan, and the United States in order to make these products available to patients with a minimum of delay. In ICH, there are 6 parties belonging to these three regions, Europe, Japan, and USA. First party is European Union, a European Commission. Second party, European Federation of Pharmaceutical Industry and Association. Third party is Ministry of Health, Labor and Welfare, Japan. Fourth party, Japan Pharmaceutical Manufacturers Association. Second last party is US FDA. Last party is Pharmaceutical Research and Manufacturers of America. Objectives of this conference is to make more economical use of human, animal, and material resources. Second, elimination of unnecessary delay in the global development and availability of new products or new medicine for human use. Maintaining safeguards on quality, safety, efficacy, and regulatory obligations to protect public health. There are 4 categories in which ICI deals, 4 broad categories represented by QSAM. Q stands for quality, S stands for safety, E stands for efficacy, and M stands for multidisciplinary. Quality related to chemical and pharmaceutical quality assurance. In this category includes stability and impurity testing. Second category is safety, represented by capital S, in which in vitro and in vivo preclinical studies were performed. This category includes carcinogenicity and genotoxicity testing. Next category is efficacy, though related to clinical studies in human subjects In this category, using dose response study and GCP, good clinical practices, etc. Multidisciplinary. In this category, cross-cutting topics which do not fit uniquely into one of the above categories. This is the tabular representation of all the categories already discussed in earlier slide. Overview of ICH. So one of first category consists of number of sections which deals with a specific topic. Q1A deals with the study of deals with the guidelines for the stability testing of new drugs and products. Q1B deals with the guidelines for photo stability testing. Q1C deals with the guidelines of stability testing of new dosage form. Q1D deals with the study guidelines of bracketing and matrixing designs for stability testing of drug substances and drug products. Q1E gives guidelines for stability data management. Q1F deals with the guidelines for stability data package for registration in climatic zone 3rd and 4th. Q2A gives definition and terminology used in analytical validation. Q2B gives the guidelines for to design methodology. Q3A deals with the guidelines about impurities in new drugs. Q3C deals with the guidelines, impurities guidelines for residual solvent substances. Q4A gives the guidelines about pharmacopoeia. Q4A gives the guidelines on pharmacopoeial harmonization. Q5A gives the guidelines for viral safety evaluation. Q5B gives the guidelines about genetic stability of the new product or molecule, pharmaceutical molecule or product for human use. Q5B give the guidelines for genetic stability. Q5C give the guidelines for stability of biotechnological product. Q5D give the guidelines for cell substrates. Q6A gives a specification test procedure and acceptance criteria for new drug substances and products. Q6B gives guidelines for specification test procedure and acceptance criteria for biotechnological and biological products. Q7A gives the guidelines for GMP for API. Q8 gives the guidelines for pharmaceutical development. Q9 gives the guidelines for quality risk management. Q10 gives the guidelines for pharmaceutical quality system. Next category is safety. Like quality, safety is also classified into different sections. S1A guidelines on the need for carcinogenicity study of pharmaceutical. S1B testing for carcinogenicity of pharmaceutical. S1C dose selection for carcinogenicity study of pharmaceutical. S2 give guidance on genotoxicity testing and data interpretation for pharmaceutical intended for human use. S3A note for guidance on toxicokinetics. S3B give the guidelines about pharmacokinetics. S4 give the guidelines about the duration of chronic toxicity testing in animals. S5 give the guidelines about detection of toxicity to reproduction for medicinal products and toxicity to male fertility. S6 gives the guidelines and addendum to ICS6, which gives the guidelines about preclinical safety evaluation of biotechnological derived pharmaceutical. S6 gives guidelines. S6 gives guidelines for preclinical safety evaluation of biotechnological derived pharmaceuticals. S7A give guidelines for safety pharmacological studies for human pharmaceutical. As 7B is the guidelines for the non-clinical evaluation of the potential for delayed ventricular repolarization by human pharmaceuticals. S8 gives the guidelines for immunotoxicity studies for human pharmaceuticals. S9 gives the guidelines for non-clinical evaluation for anti-cancer formulation. Third category is efficacy. E1, the extent of population exposure to assess clinical safety. E2A for clinical safety data management. E2B for maintenance of the ICH guidelines on clinical safety data management. E2B gives the guidelines for the revision of the ICH guidelines on clinical safety data management, as well as data elements for transmission of individual case safety reports. E2C, clinical safety data management, periodic safety update reports for marketed drugs. E2D for post-approval safety data management. In this section, we give gives guidelines for definition and standard for expedited reporting. E2E gives guidelines for pharmacovigilance planning. E2F gives guidelines for development safety update report. E3 gives guidelines for structure and content for clinical safety, clinical study reports. E4 provides the guidelines for the dose response information to support drug registration. E5 provide the guidelines for ethnic factors in the acceptability of foreign clinical data. E6 provide the guidelines for GCP. E7 provide the guidelines for studies in support of special population, especially geriatric, means senior citizen population. E8 provide the general consideration for clinical trials. E9 provide the guidelines for statistical principles for clinical trials. E10 provide the guidelines for choice of control group and related issues in clinical trials. E11 clinical investigation of medicinal products in the pediatric population. E12 provide the guidelines for the principle for clinical evaluation of new antihypertensive drugs. E14 provide the guidelines for the clinical evaluation of QT and QTC, interval prolongation and pro-arrhythmic potential for non-anti-arrhythmic drugs. E15 provide the definition for genomic markers, pharmacogenomics, pharmacogenetics, genomic data, and sample coding categories. E16 provide the genomic biomarker related to drug response. Last category is multidisciplinary guidelines, M1 for medical terminology, M2 for electronic standard for the transfer of regulatory information, M3 for clinical, non-clinical safety studies for the conduct of human clinical trials and marketing authorization for pharmaceutical. M4 provide the guidelines for the common technical documents. M5 provide the guidelines for data elements and standard for drug dictionaries. Impurities in new drug substances. This section is covered under the section Q3A. One term which is frequently used, preamble. This document is intended to provide guidance for registration application on the content and qualification of impurities in new drug substances produced by chemical synthesis and not previously registered in a region or member state. It is not intended to apply to new drug substance used during the clinical research stage of development. The following type of drug substances are not covered in these guidelines. For example, biological and biotechnological product, peptides, oligonucleotide, radiopharmaceutical, fermentation product, and semi-synthetic product derived therefrom, as well as herbal products and fruit products of animal or plant origin. Impurities in new drug substances are addressed from two perspectives. First one is chemical aspect and second one is safety aspect. Chemical aspect include classification, identification of impurities, report generation, listing of impurities in specification, and a brief discussion of analytical procedure. Safety aspect include specific guidance Guidance for qualifying those impurities that were not present or were present at substantially low levels in batches of a new drug substances used in safety and clinical studies. Classification of impurities. Impurities can be classified into following categories. First one is organic impurities, second one is inorganic impurities, and third one is residual solvents. Organic impurities can arise during the manufacturing or storage of the new drug substances. They can be identified, may be unidentified, may be volatile or non-volatile, and include starting material, byproduct, intermediate, degradation product, reagent, ligands, and catalyst. Inorganic impurity can result from the manufacturing process. They are normally known and identified and include reagents, ligands, and catalysts, heavy metals or other residual metals, inorganic salts, other material like filter air, charcoal, etc. Solvents are inorganic or organic liquids used as a vehicles for the preparation of solution or suspension in the synthesis of a new drug substance. Since these are generally of known toxicity, the selection of appropriate control is easily accomplished. Extraneous contaminants that not occur in new drug substances and more appropriately addressed as a GMP issue. Impurities may be present in their polymorphic form, cannot be identified. May be present in enantiomeric form, also cannot be identified. Reason for the reporting and control of impurity. Organic impurity, the actual and potential impurity most likely to arise during the synthesis, purification, and storage of the new drug substances. The chemical reaction involved in the synthesis impurity associated with raw material that could contribute to the impurity profile of the new drug substances and possible degradation products. The structure of actual impurities present in new drug substances at a level greater than the identification threshold can be identified. If any impurity present less than identification threshold level, then the method should be proposed for their identification. When identification of an impurity is not feasible, a summary of the laboratory studies demonstrating the unsuccessful effort should be included in the application, where attempts have been made to identify impurities present at levels of not more than the identification threshold. It is also useful to report the result of these studies. Inorganic impurities are normally detected and quantified using pharmacopoeial or other appropriate procedure. Carryover of the catalyst up to the new drug substance should be evaluated during the development. The need for inclusion or exclusion of inorganic impurities in the new drug substance specifications should be discussed. Solvents. The control of residues of the solvents used in the manufacturing process for the new drug substance should be discussed and presented according to the ICH Q3C guidelines for the residual solvents. Chemical development study conducted to scale up optimization and validate the manufacturing procedure for a new drug substances. Enantiomeric property, impurity, a compound with the same molecular formula as the drug substance that differ in the spatial arrangement of atoms within the molecule and is a non-superimposable mirror image. Extraneous contaminant, an impurity arising from any source extraneous to the manufacturing process. Herbal products, medicinal products containing exclusively plant material and oblique or vegetable drug preparation as active ingredients. Identified impurity and impurity for which a structure characterization has been achieved. Identification threshold, a limit above which an impurity should be identified. Impurity, any component of new drug substance that is not the chemical entity defined as the new drug substance. Impurity profile, a description of the identified and unidentified impurities present in a new drug substance. This is the tabular representation of threshold, identification threshold. If maximum daily dose is less than or equal to 2 gram per day, then reporting threshold will be 0.05%, identification threshold will be 0.10%, quantification threshold will be 0.15%. If maximum daily dose is more than 2 gram per day, then reporting threshold will be 0.03%, identification threshold and quantification threshold will be 0.05%. These are some examples of identification threshold and quantification threshold. The lower threshold can be appropriate if the impurity is usually toxic. For example, do known safety data for this impurity or its structure class preclude human exposure at the concentration present. Thank you. Thank you all the audience for your patience for listening. And in next lecture, we will discuss about the novel approaches in development, characterization, and evaluation of new herbal dosage form. Thank you to all. Thank you.